Good afternoon, everyone, and thank you for joining us for the Meeting the Standards, the Impact of Rapid Flu Test Reclassification webinar, which is sponsored by Sekisui Diagnostics. As we go through the call, please feel free to submit any questions that you have to me via the GoToWebinar chat window or via the hand icon on your control panel. Now I would like to introduce you to Scott Adams, the publisher of Repertoire Magazine.
Thanks, Hannah. Happy Tuesday, everyone. We're glad to have you on the call, and I'm excited to introduce Dr. Sally Hoibat, who's a retired director of the FDA Division of Microbiology. Sally is going to give us a presentation today on classification of flu, and again, just really want to thank Sekisui for stepping up and sponsoring this webinar. And like Annie said, please feel free to send in your questions as we go throughout. So with that, I'll turn it over to Sally, and then I'll be back at the end to read off any questions that may have come in. So Sally, take it away.
Okay, well, hello everybody. I hope this is going to be useful to everyone. I know that when we talk about FDA regulations, people kind of look the other way, but this one does have an impact on many people, and I'll try and cover a lot of those things. So let's go through a little bit of what I'd like to cover in this presentation today. I'm going to go through the scope of the reclassification of these influenza rapid diagnostics. What were the reasons for reclassification? Why did the FDA do this? What's meant by special controls? You'll see that's an important piece that's been put in with the reclassification. When's the implementation date for this new reclassification? And what are the implications of that reclassification of the rapid flu test for manufacturers, distributors, physicians, and lab facilities? It'll be mostly manufacturers and distributors today, the implications, but I will touch on physicians and laboratory facilities as well. I'm not going to cover the science and the clinical aspects of influenza infection because I'm sure everybody knows that it can be a real public health consequence, the influenza infections in the US. I mean, they are associated with up to 48,000 deaths annually in the US, depending on how severe the flu season is, and there's a huge economic burden. On top of that, there are the emergence of novel viruses, and we'll talk a little bit more about that too, because this reclassification does incorporate the emergence of novel viruses as well. So let's have a look at the scope of FDA reclassification rule. What exactly is it covering? It's covering those rapid influenza diagnostic tests where the intended use is actually where the test is detecting influenza virus antigens directly from a clinical specimen. And previously the FDA classified, and we'll talk about classification in a minute, as influenza virus serological reagents. If you notice at the bottom, I made a note that the molecular rapid influenza tests have already been reclassified as class 2 devices. So we're talking about the serological reagents now, the antigen tests, and they've been reclassified from, and that's the regulation in FDA terminology, they've reclassified from what's known as class 1 to another reg, which has been made to make them into class 2 with special controls. And we'll explain what that means in a minute. These devices, as you know, are usually visual and reader-based type of tests. I wanted to go into a little bit to explain to you what we mean by classification in terms of FDA. So FDA regulates IVDs, in vitro diagnostics, based on their risk. And what does that mean? Well, there's a mantra that says the more knowledge you have, it will mitigate or lessen the risk. And that's what the FDA is using in this risk class-based regulation of IVDs. In other words, in this little diagram, the least amount of knowledge you know, the more there's a risk. And here the risk is, of course, to a patient. So they divided up the IVDs into 3 different classes. Class 1 means there's a low likelihood of harm if, in fact, there's a false negative or a false positive result. And as everybody knows, you don't get IVDs, especially these serological ones, that give you 100% correct results. So the question here is, if you got a false result, what kind of harm would it be to a patient? Class 1 is usually classified they're low risk. Class 2 is there's a moderate likelihood of harm from a false result. And class 3 are the ones that have a significant risk if there's a false positive or negative result to a patient. And in that class, there are things like HIV and hepatitis B and hepatitis C and papillomavirus, whereas you'll get other infectious diseases in the class 1 and class 2. What do we mean by false negative? Well, a false negative in influenza can lead to overuse of antibiotics, failure to institute proper infection control procedures, and a false positive is sort of the opposite, and necessary use of antiviral therapy, and maybe a delay in giving antibiotics if, in fact, that patient has a bacterial infection rather than an infection such as influenza, which is a viral infection. Okay, so that's how the classification, that's class 1, class 2, and class 3. Let's have a look what that's an implication for a manufacturer, for example. Class 1 versus class 2 requirements for an influenza test, like these rapid tests. So currently, a manufacturer knows that their test is subject to what's known as general controls, sort of regulatory language here. It means that they have to have registered and listed their device with the FDA. They have to let the FDA know if there's any risks that go on once it's on market. And they have to also report to FDA any adverse event, which means something perhaps that's been connected to a false result or something wrong with the performance of the test once it's on the market. They are subject to good manufacturing practices, including design controls, and they have to submit what's known as a 510K, which is really a summary of performance of the device to FDA for them to review to make sure that once it's marketed, it's going to be safe and effective. The class 2 RIDTs have the same, but in addition, they are subject to what's known as special controls. So with the reclassification of these tests come the subject to special controls. Let's have a look a little more into why FDA reclassified RIDTs. And it's also a collaboration with the CDC on why they decided that, in fact, influenza diagnostics, when they're regulated under Class 1, don't always meet the needs of patients, physicians, or public health officials. And this was seen in particular during the 2009-2010 H1N1 influenza outbreak, where in fact only approximately 51% of infected patients with that virus strain were actually diagnosed correctly. And some studies have shown that the average sensitivity, that means the true positivity of RIDTs, is only about 63%. And in fact, it's even less in adults, it's about 53%. However, it is notable that the current RIDTs do have very good specificity. In other words, they don't give false positive results. And it's up to about 98% of the results are actually good results in that respect. So given that information, lots of papers published, there was a need to know how to mitigate or reduce the known risks associated with the poor performance of the Class 1 RIDTs. And of course, it's due to the viral antigenic changes that occur in circulating strains as this virus influenza evolves. And everybody knows that you have to be revaccinated every year because of those changes that are constantly going on in the influenza virus. So the FDA believes that general controls, you saw that on the previous slide, are not sufficient to reasonably assure that what's known as the safety and effectiveness of the rapid influenza diagnostic tests. And that by reclassifying them to class 2, that allowed the FDA to institute special controls to be applied to the RIDTs. And one of the added benefits here was to try and establish and maintain the minimum performance criteria for RIDTs throughout their product life cycle. In fact, it's saying that the tests that were actually cleared, approved by the FDA 15 years ago, and they worked very well at that time with the circulating viruses at that time. But over time, they were not able to pick up the new strains of viruses that were coming through and infecting people. So you'll see that one of the special controls is establishing to try and maintain a minimum performance throughout the product life cycle, which can be up to 15 years for a rapid test. Also, it was felt that this would also promote the development of new and improved RIDTs. So let's have a look at what the special controls have been put in by the FDA and what kinds of things the manufacturers have needed to look at. The first thing is that the special control to say there has to be a minimum clinical performance criteria requirement. And they have to demonstrate that by using a currently appropriate and FDA acceptable comparator method, comparing their device to those devices. Secondly, there's a requirement for annual reactivity testing and result reporting. And we'll go into each of these a little bit more in the slides that follow. And finally, a third one, provision for testing in a declared emergency or a potential emergency. And that's if a new strain comes in, like H1N1, and viral samples will be made available. So let's look at each of these. What are the minimum clinical performance criteria and the reference comparator methods that the FDA is asking manufacturers to follow? And by the way, this has to be for each type of respiratory specimen claimed, either nasal swabs, for nasal swabs, or nasal washes, etc. So each one of those claimed specimen types has to be able to fit in with this, these minimal clinical performance criteria, or demonstrate that they can do that. In terms of specificity, all influenza detection devices should show specificity with a lower bound of the 95% confidence interval greater than 90% for both flu and flu B, if they're claiming to be able to pick up those two different kinds of influenzas. For sensitivity, two different reference methods have been put in there. When you compare the tests that you have to a viral culture, and they do emphasize this has to be a correctly performed viral culture. A viral culture is sort of something that's like gardening, and you get differences between labs. So this is something that a company has to be very careful of when they compare their test to a viral culture to make sure that it's correctly performed as the reference method. And these are the criteria that they have to show in terms of performance. For flu A, the point estimate needs to be 90%, and the 95% confidence interval should be, the lower bound should be 80%. For flu B, the point estimate is a little bit lower because very often it's much more difficult to pick up flu B. It has to be 80%. And the 95% confidence interval lower bound should be 70%. You can compare the test to a molecular comparator method, molecular meaning you're picking up the nucleic acid. There are tests, rapid tests now for influenza that use this methodology. And here, because we know that probably the molecular tests are more sensitive usually, there's a lower boundary here for the comparison. For flu A, the point estimate should be 80%, and the 95% confidence interval lower bound 70%, and very similar for flu B. Eventually, the FDA has left the door open in this rule for other comparators, which may come along as technology improves or becomes different. Perhaps sequencing may be acceptable in the future. So there is the door open there for different comparator methods as we get better technology perhaps introduced. Let's talk about the annual reactivity testing and how you report the results of that. The idea here is that this will provide a process for continuously monitoring of an RID performance, which is on the market. So every year, the manufacturers of Class 2 RIDs should develop a post-market test plan. So after they've been cleared by FDA, they should have a plan to test annual reactivity testing with contemporary circulating viruses. That means whatever's circulating in that flu season or is predicted to be circulating by CDC and WHO, and to follow a standardized protocol. And in fact, this will also enable comparability between RIDTs. These viruses, these circulating viruses, will be available each year from CDC with sufficient time for manufacturers to test their already cleared assays. Also, we can look at that emerging influenza strain as special control. There will be, if there is an emergency declared for an influenza new strain, the FDA will be notifying developers when a strain or some specimens will be available, probably from the CDC. They usually get those from the public health labs as the pandemic begins, and they will give they will have to test that new strain and also put it within 60 days in their package inserts. They have committed to having the strains available for manufacturers within 30 days of the public health emergency being declared. So that will be very different from what happened with the H1N1 pandemic, and they have learned from that they do need to make available these new strains to manufacturers as early as they can. There will be a testing protocol and a proposed interpretation and presentation format that you will need to put in your package inserts or on your website, will be included with the viral panels when you get them from the CDC. So let's have a look at the current 2017 analytical panel for mandatory annual testing by manufacturers, which the CDC has been supplying. And I understand that just about every manufacturer of these flu tests has actually already tested their product with this. So that's been successful in terms of the manufacturers realizing that they had to apply for this and test their current test. And the selection of viruses is expected to be based every year on the strains that have been selected by WHO for the annual vaccine. For example, this panel actually mirrors the strains that were circulating in the last 2016-2017 flu season in the United States and the rest of the world. And this is what's predicted probably will be circulating this year. The CDC, by the way, just so you know, won't be posting any of the results generated from this panel and you'll see the expectation of those results being posted is actually up to the manufacturer themselves. Okay, so what are those labeling requirements? For the annual analytical testing results from the last 3 years since the device was cleared, those have to be added to the labeling, the package insert, in a separate section, or provided on the manufacturer's website, or perhaps they have a technical bulletin, and that has to be there by July the 1st of each year. If it's only on a website, the manufacturer should provide a prominent hyperlink in the package insert so that the end user can look that up. And if the device has not been cleared for 3 years, or perhaps as it is at the moment, there's only 1 year of this panel, then every year the annual results should be reported. So in the absence of reactivity, let's say the manufacturer finds that they're not picking up one of these panel members, then they need to include it as a limitation in the test labeling regarding the reactivity, and by saying that they do not detect a specific strain by their test. For the manufacturers, these labeling updates do not need to be submitted to FDA. And as I mentioned before, the results should be reported in either the package insert or on the website in a tabular form, and include detailed information for each of the viruses that have been tested in that panel. So what remains unchanged for manufacturers? They still have to comply with good manufacturing practice regulations. They still have to submit a 510K to the FDA for all new RIDTs, whether they're antigen or molecular, manual or reader result based. The requirement for all RIDTs to conduct clinical and analytical performance studies is still there. If you want to have an intended use in a point of care type of facility, you still need a clear waiver submission. And it's the manufacturer's responsibility to ensure reliable performance throughout the device's total product life cycle. And for that, I'm talking about from the time it's cleared to the time it's taken off the market, which could be, as I said before, up to 15 years for some of these rapid tests. So they do have that responsibility to make sure that the performance is as good as it was picking up whatever the strain of the virus is in that particular season. Implementation date of the reclassification, the final order was published in the Federal Register February the 13th, 2017. And the FDA gave manufacturers a year to come into compliance with those special controls. And so the special controls compliance date for all of the RIDTs that should be legally marketed is January the 12th, 2018. Just some information again for manufacturers. If you need to submit or resubmit because you've had to modify your device in some way to bring it up to the special controls compliance levels, this is what you would, some of the information that you would have there. You can see the regulatory class is class 2. The regulation, as we said before, is that 21 CFR 866-3328, which is influenza virus antigen detection system. And the new product code is PSV. What are the implications for distributors of these RIDTs? Well, after January the 12th, the FDA could take actions and seize any influenza rapid tests that you might be holding that do not meet those special controls. It's a low priority, but distributors should be warned to manage their inventory so that they only possess and distribute devices that meet the special controls as of that compliance date of January the 12th. Implications for physicians and lab facilities. It's obvious that some of the currently manufactured and distributed influenza antigen RIDTs, just a few, will not achieve the new special controls performance criteria, which they will have looked at by testing that panel from the CDC, and they will have to be withdrawn from the market by January the 12th, 2018. So what happens if you still possess some of those RIDTs, which are going to be withdrawn from the market? You can continue to use them until they reach their expiration date. So you don't have to throw those away, but do realize that you will not be able to reorder that particular brand. And when you're purchasing new flu RIDTs, physicians and labs should check the test labeling claims and the manufacturer's websites to see if the manufacturer has conformed with those reclassification special controls and has included the information on the testing of the panel, that annual reactivity panel, in their labeling. So why continue to use influenza antigen RIDTs? All antigen-based RIDTs that conform to the new FDA special controls requirements, they will continue to be valuable tools for diagnosing influenza because of the following. They're usually lower cost than molecular assays, and very minimum amount of equipment needed for many of them, although the molecular tests are beginning to have very small equipment needs as well. They can be used in low resource settings like rural areas. They can be used in physicians' offices or outpatient clinics. And they do, as we said, have a very good high positive predictive value. Just remember that that's only during a flu season. You should not be using these outside the regular flu season. You'll get a lot of false positives. But they do have a very good high positive predictive value. There will be improved sensitivity now with this new reclassification. It's only a short time to results, which really does help with appropriate treatment decisions. You know that you do need to give antivirals within the first 48 hours after symptoms for them to be very effective. And also, by getting better results, you're going to reduce the use of antibiotics, which is a really big topic at the moment in terms of trying to stop resistant organisms. So they're also, we mentioned, very useful during influenza outbreaks when public health labs often get really overwhelmed with clinical samples being sent into them often for nucleic acid-based testing or culture. In the H1N1 pandemic, there were photographs of public health facilities being totally inundated with thousands of samples and not really useful then, the results, because by the time that they gave the results, the infection was often over. So one thing I just wanted to add before any questions is that you can read the final rules for reclassification in the Federal Register. And as most manufacturers know, the FDA does give multiple guidance documents and help on their website for developers of rapid tests, especially for respiratory infections, both antigen and nucleic acid based. And there's also a process which is really pre-consulting from FDA called the pre-submission process, which is really to help developers get additional information that they may have, questions they may have before they take their product out for clinical evaluation. And you can look this up, you can Google, if you don't know how to get into the website, and look up those guidance documents on how to develop rapid tests for influenza. Lastly, I'd like to thank the sponsors for inviting me to give this webinar, and also to former colleagues of mine at FDA, Stephanie Axelrod and Tamara Feldblum, for their guidance on this topic. And in fact, Stephanie is the name that you'll see on the rule. If you have questions on reclassification, contact her for any things that you're not quite sure about. So thank you very much, and I'll take some questions if you have them.
Thank you, Dr. Hoivat. This is Scott Adams back with, we've got a handful of questions that have come in during the course of the presentation. So I'm going to jump right in and start asking those. If you have any additional questions, for those of you in the audience, please be submitting them while we're going through these handful that have come in already. The first one was, is there any chance that the reclass will be delayed until spring or later next year, possibly?
No, I think that's a fixed date. The FDA felt that they gave a year for manufacturers to check the performance of their tests, and that's a solid date.
Okay. The next question was, how do I know what to look for in a package insert to know if the rapid test that I'm currently using/selling meets the guidelines?
I think, as I mentioned, what the manufacturers have to do is put in their performance section or the technical bulletin or their website the information in a table format that the CDC has asked them to do and has recommended they do. So that's the responsibility of the manufacturer, and if it's not in the package insert, there should be a good hyperlink to information that may be on the web.
Gotcha. The next one's on a nasal wash. The test kit that I'm currently using has a nasal wash that passes the FDA requirements. However, it does not for the nasal swabs. Will the kit still be available to use after 2018?
Yes, potentially it would, but the manufacturer would have to remove the claim that they have for the specimen type that did not pass that performance criteria in the special controls. So you would have to use that kit for whatever is now the claim that the FDA is allowing.
Gotcha. I got an FDA question, which are always fun. Did the FDA seek the opinion of physicians before implementing this RIDT reclassification rule?
Yes, they did. They have to. That's something that it avoids criticism. When they do something like this, they have a microbiology advisory panel, which is the selection of experts in a field. And in June the 13th, 2013, that panel met to look at the recommendations of CDC and FDA. And that panel consisted of 6 physicians and 7 researchers. And each one of them was an expert in influenza infection, treatment, epidemiology. And there were also representatives from CDC and the Association of Public Health at that meeting. And they presented extensive data on the performance of the currently available, at that time, rapid influenza test, which supported the conclusions that, in general, they were poor performance. That panel recommended unanimously to reclassify RIDT. And then there was a comment period on the draft rule. This happens with all sort of guidance documents, most guidance documents, unless it's something that the FDA has to rush through. And so there was a 90-day comment period on the draft rule where the FDA did receive a lot of comments from individuals in industry and physicians, etc., to their draft rule. And they incorporated some. They also publish what comments came in and why they did or they didn't decide to take the recommendation. So that's a typical kind of thing FDA does, just to make sure that they are not just making a decision on their own and including the people who it would impact the most.
Gotcha. Will there be an appeals mechanism for manufacturers whose tests did not comply with the special controls by January 2018?
I did check with the FDA, and the answer was no, unfortunately.
No appeals.
No
So
appeals.
they're either in or out.
You're either complying with the special controls by January 2018, or you're not, and then you would be selling something which wouldn't be a good idea to sell if the FDA
Right,
wanted it.
So you're starting over with a brand new 510K, basically.
Only if you have to modify your test.
Gotcha. Why did the FDA use two different criteria for the performance standards?
That's a good question, and I believe that's one that came in as part of this comment period. At first, everybody thought, well, because of the issues of difficulty of running viral culture, that individuals wouldn't particularly want to use that as their reference method. But in fact, the viral cultures detect the viable virus particle, and nucleic acid-based methods, the molecular methods, detect viral RNA. So they're sort of detecting two little pieces here: viable virus particles with the viral culture and the viral RNA with a molecular test. And it turns out that it may be, one of these two different reference methods may be a better comparator than the other for a particular RIDT. So the FDA has left that decision up to the manufacturer to make on what they would like to compare their test to.
Let's see, I got one in. See if I can read it. Do we see this type of reclassification moving to other types of testing as we move forward in the years to come? This came in from one of the callers, especially as we move forward towards value-based care models.
Every year or so, the groups at FDA, in particular, I'm referring to the microbiology division that I used to direct, we're asked to look at that question. Can we down classify? So that's the opposite. Can we take something from that class 3 criteria, which does involve a lot more information being sent to FDA, costs a lot more for a manufacturer to do? Can we down classify that because we have more information, which minimizes the risk? And the answer is yes. There has been down classification. There was a down classification a couple of years ago of some of the TB tests that was instituted. So it went from a class 3 to a class 2 product. What we've seen with reclassifications from class 1 to class 2 is that most of the molecular assays that have gone through, at least in the microbiology division, have been, once they came in, they were elevated to class 2, just because the FDA has a little bit more control post-marketing of a product and has much more control over the performance, as you've seen, to try and maintain a good performance of a particular set. So yes, there's always a question of, can you down classify? That's usually what especially manufacturers would like to see. And if there's good reason and justification, and the manufacturers have made a good case for that, perhaps we know much more about a particular infection which lessens the risk, then that will proceed. It takes a long time, though. I mean, these things don't happen overnight. You can see that even with this up classification, the meeting was held 2013, and it really took almost 4 years for this to go through. So, I mean, that's something the FDA maybe will start to work on to try and see if they can reduce the timeline for reclassification, either down classification or up classification.
Great answer, thank you.
Yeah.
Last question here. This one looks like it comes in from the supply side. It says, our research implies up to 60% of physicians are not aware of the reclass. Is the FDA contacting them to inform them of what's going on with this program?
I don't believe so, but I think there have been, I did notice that AACC, American Association of Clinical Chemistry, and I believe the Public Health Association have also had webinars on this topic. So I think that education part is trying to be done by those associations for the clinical chemistry, the lab people, and the public health people. But the FDA did announce this, but did not, they're not really responsible for making sure that information gets out. I think this kind of webinar is what's needed, but you can also, well, I guess the physicians wouldn't be interested too much in the manufacturing side, but we are having another seminar, a webinar, I think, in about a month, which is based mostly trying to get a physicians and the end user type of person involved. So hopefully through these webinars, the message gets out. But I have heard that physicians are so busy. It's usually the point of care head person, I think, in the case of clinics, that's really the ones that have been most affected by this. And talking to my FDA former colleagues, some of the questions that are coming in are from that category of individuals. So it's the point of care coordinators who do seem to have heard about this, but have lots of questions. So they can contact FDA as well.
Lots of great information. Thank you so much. Hopefully this was helpful for the audience. It's a great way to use some content to go in and talk to your customers about some different things that will be affecting them. So hopefully this content sponsored by Sekisui was very beneficial for you. And we'll send out some follow-up emails and things regarding this webinar, just as reminders and some bullet points for you guys in the coming weeks. So Dr. Hoivat, thank you so much. Thank you, Sekisui, for sponsoring, and we hope everyone has a wonderful day. Thank you.